Okay, leaving the cochlea, we move into the auditory nerve. Here's a reasonable way to think about this. We think about the hair cells in the cochlea as being mechanoelectric transducers. And if you want to compare them to devices that you're more familiar with, you can think of those as microphones. That is to say, the acoustic energy from somebody's voice moves around the diaphragm, moves around the different elements of a microphone, physically moves them, and in doing so, the energy of the voice is converted into an electrical signal that can be passed through a wire into a computer or a public address system or something like that. And when we now take the cochlea uh, uh, microphone comparison one step further, we would acknowledge or we would recognize then that the auditory nerve is the wire that carries that electrical signal from the transducer, from either the microphone or the cochlea, into the central nervous system. In the hearing business, that nerve fiber, or I'm sorry, that nerve is the eighth cranial nerve or the vestibulocochlear nerve. Sometimes it's called the acoustic nerve. But in any case, it's the eighth cranial nerve. And again, if we look at the cochlea as a very fancy microphone, we look at the auditory nerve as a very fancy wire carrying energy from that microphone into the central nervous system. As I look at this slide and I see eighth cranial nerve there, I begin to think of potentially a makeup assignment or some kind of extra credit assignment whereby the students who would be interested in memorizing and reporting on the cranial nerves, there are 12 of them, reporting on the cranial nerves, we could perhaps work something out there. Now, I don't want you to just sit there with a book and write down what's written in a book. This would actually have to somehow demonstrate that you all had memorized those nerves and their function, but maybe that's something we can think about for the next week or two here, as I know many of you are looking for an opportunity to support or improve the grade that you've got following the quizzes and the midterm. If you are worried about that, believe me, you're not alone, and we'll try to do whatever we can to get everybody an opportunity to show what they know in the class. Okay, in any event, just needed to get that off my chest, sorry. In any event, the eighth cranial nerve has two portions, which would make sense, right? Because the inner ear contains the cochlea and the hearing section, if you will, and there's also the vestibular or the balance portion. And the nerve is actually divided up into these two divisions or these two branches. The so-called pars inferior, the inferior part, I guess you could think of it as, which serves the sensory cells in the cochlea and the saccule. The saccule is actually one of the vestibular organs, but its nerve fibers, its associated nerve fibers travel with the cochlea, the fibers from the cochlea. And then there's the pars superior, which is charged with communicating with carrying the signals from the sensory cells that are in the rest of the vestibular system, specifically the semicircular canals and the utricle. Here's a nice little image of that where you can see here the cochlear branch of the nerve. Here's the saccule, and so its nerve fibers would ultimately kind of hook up with those from the cochlea to form that so-called pars inferior portion of the nerve. And then here are the semicircular canals, these guys right here, and the utricle is this organ right here. And these mechanisms have their own branch of the eighth cranial nerve, their own branch that serves the vestibular system. And as you can see, it meets up with and ends up traveling with the rest of the fibers, those coming from the cochlea and the saccule, to form the eighth cranial nerve in its entirety. So there's these two branches, the cochlear nerve and the vestibular nerve, that together form the vestibulocochlear nerve. I want to point out one other thing here. If you look at this part of the diagram, you see how the cochlea, the coils of the cochlea would be kind of going off into the screen. And each one of these little lines right here represents one of those nerve fibers. There are about 30,000 of them altogether. And you can see how they kind of emanate out from the cochlea, from the coils of the cochlea, and wind together to form the trunk of this nerve. Now, this area right here would be the base of the cochlea, the basal portion of the cochlea. As you go deeper into the screen, you would be heading toward the helicotrema. You'd be heading toward the apex of the cochlea. And I want you to see how these fibers here actually kind of lay down on the outer portion of the nerve. So just like the cochlea has tonotopic organization, so does the nerve that communicates with the hair cells in the cochlea. That is to say, the nerve fibers from the base of the cochlea rest on the outside portion of the nerve. These fibers in here then would travel all the way through the coils of the cochlea, making it all the way up to the apical portion of the nerve. So the fibers that go to the apical portion of the cochlea are actually longer than the fibers that go to the basal portion. And also, the fibers that go to the apical portion form the inside part of this nerve, whereas the fibers from the basal portion, which you see right here, form the outside portion of the nerve. Now again, this is just the hearing part of it, but for our purposes in this class, that's where we're going to be, of course, focusing our attention. Okay, so the nerve supply to the inner ear, just like the nerve supply to the rest of the body, there are ascending and descending pathways. Ascending, typically thought of as sensory pathways, in that there is some activation that occurs somewhere in the body or somewhere in the head and neck region or through one of the senses, and then that energy is converted into nerve impulses that then go up rostrally through the central nervous system, through the brainstem, eventually up to the cortex for processing. And you think of that as moving up, rostral, toward the head, and therefore ascending. The official name for that would be the afferent pathway or the sensory pathways. And then there are descending pathways as well. These are going to be the nerve pathways that allow your brain to activate different muscle groups throughout the body. Here now the neural commands rather than, or neural signals, rather than originating somewhere in the periphery, somewhere on the sort of the outside of the central nervous system or the outside of the body, now the nerve impulses for this pathway would be originating in the brain and would be descending through the central nervous system out to the rest of the body. Excuse me, the official name there is the efferent pathway, and you can think of that as a motor pathway. So afferent, ascending, sensory, those all go together. Efferent, descending, motor, those all go together as well. We have already mentioned that it is the eighth cranial nerve that activates or innervates or communicates with the structures in the cochlea, specifically the hair cells. And again, we've already mentioned the afferent divisions of the eighth cranial nerve, the vestibular branch and the auditory branch. And as we'll talk about in a few minutes here, it turns out there's also a small efferent bundle of nerves that travel with this pathway, specifically with the vestibular branch. And this small collection of nerve fibers actually is what provides the electrical stimulation for outer hair cell electromotility, which you all saw hopefully as the dancing hair cell in the previous set of animations. So remember that outer hair cell was moving, and remember the inner hair cells don't, only the outer hair cells do this. The outer hair cells moved when stimulated electrically. That process is called electromotility. We also talked about it in terms of it being electromechanical transduction, in that an electrical signal produces movement, produces some kind of outer hair cell movement and changing of shape. So it is these efferent fibers that facilitate, to some extent anyway, the movement of the outer hair cells when the outer hair cells are stimulated electrically. So, okay, fine, that sort of sets up the eighth cranial nerve. Now let's back up just a little bit and take a a look at how the nerve fibers are constructed. Some of this should hopefully be a review for some of you because this is high school biology stuff to some extent, but let's make sure that we're all calibrated here. We think of neurons as having three main parts: the cell body, which is where the nucleus of the neuron is. The nucleus of the cell resides in this cell body. A neuron, by the way, is just a nerve cell. We also think of the cell body as, in terms of it being the decision maker for a nerve cell with respect to whether that nerve cell fires or not, whether that nerve cell actually gets stimulated enough to discharge a nerve impulse. I don't want to get too far ahead of ourselves here because we're going to talk more about nerve impulses in the next lecture, but just recognize neurons make decisions with respect to whether they are going to fire nerve impulses or not, and that decision is made in most cases, for the most part, in this cell body. Now there are also projections off of the cell body, and these are called neurites. And you can think of a nerve cell as having an input side and an output side. The dendritic surface is the so-called dendritic surface is the input side. So you can think of the dendritic surface as sampling the environment and, in a sense, allowing the environment to interact with the neuron in such a way that the neuron either will fire or not. But in a sense, the dendritic surface is the cell's sensory surface. The output side of the neuron is the axon or the axonal surface, and it's kind of similar to an actual wire or cable that's going to carry an electrical or a chemical signal away from the cell body, typically toward another neuron. So if you want to think of neurons as talking to each other, you would think of the dendritic surface as the side that's listening to the environment, the cell body determining what's happened and what the response ought to be, and then if a response is required, it would be the axonal side of the neuron that would deliver that response to wherever the response needs to go. So you've got dendritic surface as the input side, cell body as kind of a calculator, determining what the response ought to be, a decision maker, and then an axonal side, which is the output side of the neuron. Here's a pretty simple diagram of one where you've got the dendritic surface over here, many branches, lots of surface area. You've got a cell body right here, and then an axon, an output side, an axonal surface that will allow for communication down here to other neurons whose dendritic surfaces, if you will, would be connected to the axon endings. Now, of course, they don't really look like that. Here's more of what a neuron would actually look like. You see there are neurites kind of going all over the place. It's a much more complicated situation than this. And it's worth recognizing that when you think of a network, when you think of something like, for example, Facebook, it's really a lot like a neural network in that there are a bunch of connections, some of which are reinforced through repeated communication, others which are not. But to me, this looks a lot more like what Facebook would look like if you kind of, stripped away all the artifice and just looked at the actual connections between people. Some would be much stronger than others. So, for example, your tendency to communicate with a friend of yours would reinforce this contact, if you would. It would reinforce this pathway much more than your communication with me, which would be a very weak, underutilized pathway. In no way is that an indication that all of you should try to friend me on Facebook. You should know ahead of time that I very rarely look at it. Just because who cares? I just don't do it. Anyway, so there are different kinds of neurons in the sense that the orientation between dendrite, axon, and cell body is not identical across neurons. The ones that we deal with the most, I think it's safe to say, would be this kind right here. Again, where you've got a dendritic surface that, in a sense, collects information from across a wide surface area and then transmits it down the axon. Bipolar neurons are also fairly common in sensory pathways. Again, dendritic surfaces, more or less, sending signals to a cell body that then decides yes or no to propagate a nerve impulse down the length of that axon. In the central nervous system, now we're kind of getting out of a specific sensory system, but there are, again, many different types of neurons. These pyramidal cells are crucial for motor activity, getting their name, of course, from the sort of pyramid shape of the cell body. There are different kinds of cells that exist in the cortex, cerebellar cortex, others in the cerebral cortex. Lower motor neurons would be the neurons that go from the central nervous system out to your limbs, for example, away from the spinal cord. These pyramidal cells are kind of interesting, though, and you can see they're much more complicated than you would be led to believe by this simple diagram there. And when you put them in a brain with other pyramidal cells and other cell types, you see just how networky this starts to look, just how many connections there are, how in some areas the connections are very dense, in other areas they may not be quite as dense. Sometimes it is referred to as a forest, in addition to being referred to as a network, again, because of the complexity and the intertwining of the various neurites, the intertwining of the projections from these cells that facilitate communication with other nerve cells. The vast majority of the neurons in the auditory pathway are sheathed in, they are insulated by a layer of fatty tissue called myelin. And myelin arises from a specific type of cell called a Schwann cell. Right there's the spelling of Schwann. And this fatty kind of insulating material actually serves to speed up the velocity with which nerve impulses will be propagated through these fibers. So there, as we'll see in a moment, there are different kinds of nerve fibers. Some have myelin and some don't. Again, as I just said, the vast majority of the fibers in the vestibulocochlear nerve are myelinated. And the significance of that is that the nerve impulses that travel through these nerves travel much faster, and then when you compare across fibers, in a much more coordinated manner than they would travel through fibers that were not myelinated. So when you hear the term myelin, what that refers to is the actual sheathing, if you will, the insulation around the nerve fiber itself. So again, here's the nerve fiber. This is the axonal portion of the nerve. You can see because it looks kind of like this, right? And it's sort of like what we've done, I'm sorry, is we take a cut right there and then look at the inside of it, and we'd see this would be the nerve fiber itself covered with, sheathed in, insulated from the environment by the myelin, which is produced by a Schwann cell. You see this little opening or this little separation between these two Schwann cells. Here's one Schwann cell, here's another. And this node of Ranvier, or Ranvier, I guess you could say, but you know, if you're feeling French, I guess you'd say Ranvier. But this opening here allows for the flow of charged particles, ions, from the extracellular area, from the area outside the cell, into contact with the axon. And we'll see the significance of that again in the next lecture when we talk about the propagation of nerve impulses from one nerve to another. So here's an example then of, again, a myelinated nerve cell, and then this would be one that does not have myelin around it. And you can see the nerve fibers themselves do not contain this rather effective insulating material around it. The main difference between these two nerve fibers would be the speed with which an action potential moves from one location to another. Action potential just being another name for nerve impulse. Now, the other thing I want to draw your attention to in this diagram is the packaging if you will, of the nerve fibers into these bundles. So there will be a bunch of nerve fibers, and typically it would be one type or another. You wouldn't necessarily see both types traveling in with, kind of one package there, but sometimes you do. In any event, you see how these nerve fibers would be packed with a bunch of other nerve fibers and then covered in a membrane called the endoneurium. And then a bunch of these endoneurium units, here's one, here's another one, here's another one, here's another one, they would be packed together. They would be packed together in these units of perineurium. So let me rephrase that again. The endoneurium is the area inside here where all the nerve fibers are. They're packaged up inside this membranous sheath called a perineurium. So bundles of nerve fibers like this one and this one would be what's known as the endoneurium, or referred to here as the endoneurium. And then these bundles of nerve fibers would be packed together in membranes called the perineurium. These packets of bundled nerve fibers, if you will, would then be bundled together themselves in yet another layer of tissue called the epineurium. What's the reason for this? What's the value in this? Well, imagine if you've got a problem that arises in the nerve that is a degenerative disease or some kind of trauma, and it eats away at part of this sheathing here. All these other nerve bundles are going to be spared the damage because they're all packaged up independently of the few of them over here that might have gotten damaged. So in this way, the spread of damage or the spread of the effects of a trauma through the nerve trunk, through the nerve bundles themselves, will be controlled a little bit. It will be minimized because there's so much, if you will, packing material protecting the individual fibers throughout the nerve. And, that makes a lot of sense. These nerve fibers are important, and the work they do is extremely important, obviously. And so their protection is going to maintain function not only of motor systems, the muscles in the body, but also the the sensory systems, the afferent pathways as well. Okay, so here's a nice diagram again of a neuron where you can see the sort of large surface area covered by that dendritic area. You can see the cell body, and then you can see the axonal projection out with the axon terminals that will communicate with dendritic material from yet another neuron somewhere over here, and also the packaging, if you will, the insulation of myelin around that axonal projection. Those nodes of Ranvier that I just mentioned would be these little spaces in between here. Their significance, again, will be reinforced on the next lecture. Just so you get a picture of, again, what some of these nerves look like when they're kind of in situ, if you will, in the situation which they're normally found. So these are neurons in the hippocampus. You see a bunch of different cell bodies. That's the little bulges right there. And you see these dense connections, if you will, between different neurons. The hippocampus is one of the memory storage units in the brain. It's specifically in a part of the brain that is associated with your survival instinct. And so you can think of the value of having these kinds of connections. Just think for a moment of how the environment, for example, can resemble or can appear threatening to you. And usually the environment will appear threatening to you when it resembles something that you remember being threatening. So you are walking down a dark street on a rainy night somewhere alone in an unfamiliar neighborhood, and maybe something happened to you before, or you remember hearing something about something happening to somebody in the past, and that kind of memory would trigger a lot of activity potentially in this network, which would then perhaps, hopefully, provide for you memories of coping strategies or memories of ways that you could deal with that kind of stress. So we want to value the connections between neurons, and we'll talk more about those in a few minutes, the actual connection point between neurons, because that is what is going to facilitate the sending of, the transferring, if you will, of nerve energy from one part of the central nervous system to another. And it will be the movement of these chemicals, the movement of these electrical signals that provoke in the brain specific perceptions and ultimately responses. Once we get into the central nervous system, once we get into the brain, there are a variety of different cell types, as I've indicated before, but some of these are actually kind of just like scaffolding. These glial or these support cells actually serve kind of as scaffolding for the nerve cells, which, although they're hooked up to each other in these networks, still require some support to maintain their shape and maintain their connections. So you can think of these glial cells or these support cells kind of like the infrastructure upon which the nerve cells send their projections to one another and, of course, maintain their distance from one another, which ultimately is important for their normal function. All right, so let's break down these different parts again with a bit more detail now. There's the cell body. Again, that's sort of the decision maker in the cell, as well as the storage space for all the genetic material of the cell. And inside this cell body are a variety of structures, some of which you might remember from, again, from biology classes: the mitochondria, the Golgi complex, the different parts of the cell body that actually maintain the cell's metabolic activity and in doing so keep the cell healthy, alive, and able to discharge nerve impulses when necessary. Now, to do that, there are a lot of different kinds of, almost like a circulatory system inside the cell, these microfilaments and microtubules, which transport a variety of chemical compounds, nutrients throughout the cell. The mitochondria generally are credited with using metabolic activity to generate energy for the cell. So the mitochondria would be, in the parlance that you might remember, the powerhouse of the cell. And as we move on, we recognize that all of this is contained within usually some kind of bilipid plasma membrane, a fatty membrane that is semi-permeable. It allows some things in, it allows some things out, but the cell body itself, the decision maker, if you will, of the cell, will at times make the cell more or less permeable to a variety of different chemical compounds or ions. So we think of the cell membrane as being its wall. It separates the intracellular fluid from the extracellular fluid. That is to say, all the stuff inside the cell would be considered part of this intracellular material or fluid. And then everything outside the cell would be extracellular. Now, keep in mind that the vast majority of our bodies is composed of salty water. So this extracellular fluid is, for the most part, salt water. And we need to maintain separation between that salty water and the fluids inside our cells if we are going to be able to do the kind of work we need to do to function specifically with respect to processing sensory information or motor activity. And of course, a human being whose intra and extracellular fluids are allowed to combine completely will not be able to carry out sensory or motor activity. That person would actually be a cadaver and not a living person. So we spend a lot of our energy just being alive. A lot of our energy is spent keeping these two fluids separate. The cell membrane facilitates that, but as we will see, there's a lot of energy that goes into the maintenance of these two different kinds of fluid. The ion transport, to which I referred just a moment ago, really involves, primarily for our level of understanding at this point, excuse me, the ions potassium and sodium. These two ions, of course, they're just atoms. They're not even molecules. They are atomic charged particles. So as you can imagine, they can slip in and out of cells pretty easily. For the most part, neurons spend most of the time evacuating sodium from inside the cell and pushing it back out of the cell, even as potassium is essentially allowed to pass pretty easily. The importance of this, again, will become clear later when we look at the generation of what's known as the action potential. Again, action potential is just another name for nerve impulse. What about the dendritic surface? Well, again, that's the input side of the neuron. That's the side of the neuron that is going to, in a sense, collect material, collect neurotransmitter, collect ions from the environment, and inform the cell body of what's out there, in a sense. The dendritic surface typically has more profuse branching than the axonal surface. And the dendritic surface is going to conduct chemical or electrical signals toward the cell body. In a way, you can think of this as the dendritic surface as an extension of the cell body itself. So again, this image captures that nicely. Here's the cell body, and you see how the cell body is, in a sense, spread out a little bit, therefore able to sort of sample the environment more effectively when you consider it as occupying this dendritic surface. And again, I think that is illustrated here really well. So the dendritic surface is going to be responsible for propagating energy, sending energy in toward the cell body. Oh, look at that, I had it right there. Okay, then there's the axonal, the output section of the cell body. Again, that's going to be this part here. And this is really the thing that we tend to think of when we think of what a nerve looks like. When you think of a nerve, you think of a wire, and that wire is really the axonal projection. By the way, this term process just means projection. So if you think of the axon as a cell process, you're really just calling it a projection. And it projects away from the cell body and ultimately conducts nerve impulses away from the cell body. There are different sized axonal projections. The larger they are, the faster they transmit their energy. Sometimes they have branching sections that go all over the place. Again, we're not going to worry about that right now. And the axon terminal, which is this area here, is going to be the area that is responsible for conveying whatever chemical messenger this cell is providing, conveying that messenger onto another neuron. And it will convey that energy or that signal onto the dendritic surface of the next neuron, which of course would be somewhere over here. The axon hillock, which is this section right here, the very first segment of that axon as it projects off the cell body, that's where the nerve impulse actually gets its start. And again, we'll come back to that for the next lecture. Now, the thing that I wanted to talk about next, though, is the connection point between neurons, known as a synapse. This is the actual physical location where you would have an axon terminal from this neuron communicating with the dendritic surface of another neuron. And the direction for this communication, if you will, is typically unidirectional. That is to say, this axon does not act like a dendritic surface. There's no input here. There's only output from here going somewhere else. This dendritic surface that would be communicating with these axon terminals would then carry energy into, or some kind of signal into, a different cell body. So the transmission is always from this side to this side, not in reverse. And by implication then, the action would always be going from this neuron out here to the next neuron, and not from this neuron back through this neuron here, unless this neuron over here had an axon that connected up with this one over here. And that kind of thing can happen, but for the most part we think of this as a evidence of a unidirectional or single directional communication. And as I indicated before, the communication, if you will, between these neurons will either be the transferring of chemical or electrical signals. And for the most part, the neurons in our bodies are going to use chemical messengers. There are different kinds of synapses. Axodendritic is the one that we would think is most common. That's the one we've been talking about so far, where you've got an axon communicating with a dendritic surface up here. You can also have axosomatic. So imagine now that these axon terminals actually fit onto the cell body of a different neuron over here. Or you can have axoaxonic, where these would actually fit onto the axon of another nerve fiber or another nerve cell, and that would, again, allow for the transfer of these chemical signals, but just in a more unusual way. With respect to the synapse, the actual separating point, the actual location where the two neurons will communicate with one another, is across a very narrow space that physically separates these two neurons, and that's known as the synaptic cleft. So when you talk about neurons actually communicating with one another, what they really do is send chemical signals to one another across a very narrow space. So here's the synaptic cleft. Excuse me. This would be the axon terminal from a neuron that's over here. And this would be the dendritic surface of a second neuron that's over here. So if we look at the if we look at the synaptic cleft as the place where the action is really going to occur, then we have a presynaptic neuron and a postsynaptic neuron. So we go from a presynaptic neuron over here to a postsynaptic neuron that would be over here. Conversely, there probably would be a presynaptic neuron over here somewhere, and then this would be the dendritic surface of the postsynaptic neuron. So this area here, in that case, if it's postsynaptic, would be this part here. And if this was the presynaptic neuron, then the axon terminals over here would be located here. And what you see is that there are going to be these chemicals that are transported across the synaptic cleft into this postsynaptic neuron. And they're either going to be accepted through this membrane or kept out by this membrane. So first neuron, if you will, presynaptic neuron. Second neuron, if you will, postsynaptic neuron, separated by this very narrow channel, the synaptic cleft. This video that I've got posted here, I'm not going to play it for you now, but I encourage you to take a look at it. There are a couple others in the slides that follow that are really pretty nice animations of what is going on as the neurons send these chemical signals across the synaptic cleft. Here's a fairly large, if you will, relatively speaking, postsynaptic neuron. So here's the synaptic cleft right here. And really what you see are these actual locations where the cleft actually is kind of active. And what you see are these little vesicles, these little packets of chemicals that are going to go across the cleft, just as you see in this drawing here. These little packets of chemicals that are going to come down when the cell is stimulated, come down to this presynaptic membrane and then dump out into the synaptic cleft, where they will then be either taken up or rejected by this postsynaptic membrane. So again, here they are again, just from a photograph rather than a drawing. And they would cross in these areas here. Look, there are several neurons hooked up to this one neuron right here. And that's not at all uncommon, where you'll have one dendritic surface receiving inputs from several other neurons simultaneously. That's not at all uncommon. Just take a look, though, at how these neurons are going to be actually physically sending bundles of chemicals. And again, each one of these little circles here, hopefully you can see them pretty well, represents one of these little packets, vesicles of filled with neurotransmitter and chemicals that will then provoke in this postsynaptic neuron some kind of response. Okay, so we have these nerve fibers, we got these synapses, all that stuff. How does this actually fit in with the cochlea? How does this actually go along with what we've already been looking at? Well, by now this should be a fairly familiar sight to all of you. Hopefully, here's the medullus, here's the eighth nerve as it travels through the medullary region of the cochlea. All of this is temporal bone out here, scala vestibuli, media, and tympani. Here's the organ of Corti. There's your 3 outer hair cells. There's your 1 inner hair cell. And what we're going to look at now is how these nerve fibers actually go through the bone and synapse onto these hair cells. So we got outer hair cells synapsing onto fibers, and we got inner hair cells synapsing onto fibers. There's a little hole in the bone right there, and these fibers are going to go through that bone, gather together in what's known as a ganglion, which is a collection of cell bodies in the peripheral nervous system. And this ganglion in the cochlea is called the spiral ganglion, as you can imagine, because as you should be able to picture by now, it spirals around with the coils of the cochlea, spirals around the medullar region of the cochlea. These are afferent, for the most part, afferent fibers, sensory fibers. And again, one other thing you can see here is that if this is sort of from the basal turn of the cochlea, you see how these fibers are on the outside of the nerve, whereas these fibers over here from a higher up coil would actually be forming more of the trunk, the inner part of the nerve, rather than this outer part of the nerve. Another image of this, nice drawing right here, I think, where you have nerve fibers, and you can see, wow, there are several of them that go to these inner hair cells. Here again is your reticular lamina. Here again is your tectorial membrane. It's not, this sort of chopped up little thing, but rather it's a continuous membrane that goes all the way through the coils of the cochlea. We've pulled it off of these hair cells right here to expose the stereocilia. And what we see is that the majority of these nerve fibers actually seem to go to the inner hair cells rather than the outer hair cells, and more on that later. But they do have to cross through the organ of Corti to get to the outer hair cells. And so there's going to be some significance to that as well. But for now, it's important to recognize that the majority of these nerve fibers actually synapse onto inner hair cells rather than outer hair cells. The bone, that is to say the osseous spiral lamina, you see one piece of it there, and here's another piece of it right here, actually has a series of very small holes in it, very small perforations called the habenula perforata. And it is through these perforations that the nerve fibers are actually able to get out from the bone, and synapse onto, make contact with the hair cells of the cochlea. Here's another image of it. Here's your inner hair cells. Here's your outer hair cells. Stria vascularis, Reisner's membrane. Here are those nerve fibers traveling through the bone on the inside of the skull and making it through these little perforations to have contact with, to synapse onto the hair cells of the cochlea. Turns out that of the 30,000 fibers that make up the auditory nerve, 19 out of 20, 95 or so % of those fibers actually go to inner hair cells. That's right, inner hair cells. So even though there are 1/3 as many inner hair cells as outer hair cells, those inner hair cells actually receive 90 to 95% of the afferent, that means the sensory fibers that compose the auditory branch of the vestibulocochlear nerve. Think about that. That means that 1/4 of the overall hair cell population, one row of inner hair cells, transduces 90 to 95% of all the nerve energy, and I guess what I should say is transduces 90 to 95% of all the hydraulic energy in the cochlea that will ultimately be sent through nerve fibers to the brain. That is stunning. That is exactly the opposite of the way you would think it would go, isn't it? You would think the outer hair cells are much more numerous. They must be the ones that are more important for hearing because there's 3 times as many of them. In fact, it is the inner hair cells that are responsible for 90 to 95% of the transduction that will result in afferent signals going to the brain. The outer hair cells, by contrast, travel through, across that organ of Corti, technically across that tunnel of Corti. You see it going there, the tunnel of Corti, right? And wind their way to the 3 rows of outer hair cells. And you see how while there are many fibers going to one inner hair cell, one fiber that goes to the outer hair cells actually goes to many different outer hair cells. So you can think of the inner hair cells as producing a divergent pattern of innervation. That is to say, the inner hair cell activity diverges to many different nerve fibers, or conversely, many nerve fibers converge on one inner hair cell, whereas one nerve fiber diverges to many different outer hair cells, or that is to say, the activation of many hair cells converge onto one fiber. This difference between type 1 and type 2 fibers, specifically, these are myelinated, these are not. And that makes sense. These fibers have to fit into a smaller area. They got to go through a tortuous little track here to get to these outer hair cells. If they were myelinated, they wouldn't fit. These inner hair cells are able to fit more easily with their myelin because they're all going to the same place, basically. So, clearly, if you lose all your inner hair cells, you lose 90 to 95% of all the afferent energy coming out of the ear. That is profound deafness. If you lose all the outer hair cells, you still retain a lot of usable hearing. Sure, your ears won't work as well as they should, but because you're only missing now 5 to 10% of all the afferent signal, yeah, you can still do okay. The bigger loss when you lose the outer hair cells is losing that electromotility, losing that electromechanical transduction that makes the traveling wave so much more effective. So in summary, here's what we see with respect to the 2 different fiber types. Now, I also said there was this efferent hookup. And in comparison to the afferent hookup, it's really small potatoes, but it is there. And so here we see a magnified version of the organ of Corti. Here's the tunnel of Corti. Here's the outer, the fibers that are going to go to those outer hair cells, illustrated in red. We've got the efferent fibers going through this system as well. In fact, I think in this diagram, the efferent fibers are the red ones, and the green ones are the afferent fibers, and it doesn't really matter in the sense that you see how they both synapse onto the base of these hair cells. But look what happens here on the inner hair cell side. This blue one is like one of these, and it is the afferent fiber synapsing onto the bottom or the base of an inner hair cell. And the efferent fiber is hooked up not to the cell itself, but to the neuron that is synapsed onto the hair cell. And it turns out that the function of these hookups is actually very different when it comes to comparing the outer hair cells from the inner hair cells. On the outer hair cell side, the efferent signal is credited with producing electromotility, or that electromechanical transduction. Specifically, when the hair cell is stimulated, it dances, just like you saw in that video. On the inner hair cells, though, the efferent nerve ending hooks up to the afferent nerve endings of the 8th nerve and actually has an inhibitory effect. So when an efferent nerve fiber synapsed onto one of these afferent fibers for an inner hair cell is activated, it actually inhibits the activity of these afferent fibers, in a sense kind of turning down the activation. So whereas here we get this nice excitatory effect, here we get an inhibitory effect. What in the world would you want that for? Well, suppose that the efferent signal could have some kind of suppression of background noise type effect. That would mean basically that that some inner hair cells respond more to background noise than others. In our world, the low frequency sounds that we hear in the environment tend to be more background noise, tend to be less meaningful than the higher frequency sounds. And it turns out that these efferent fibers on the inner hair cells have an inhibitory effect that is stronger for low frequency sounds than for high frequency sounds. It's almost like we have a built-in noise suppression system that facilitates our ability to distinguish signals we want to hear from background noise. If that idea is of interest to you, I do suggest you consider a career in audiology. All right, just to kind of wrap some of this up now and to put it all back into the central nervous system, we've got these ascending and descending pathways. We've got these afferent and efferent systems, which are respectively sensory and motor. And we also need to recognize that there's not only a central nervous system, but also a peripheral nervous system. And the naming of some of these structures, these different these different nervous tissue structures is going to depend upon where they're located. We're also ultimately going to talk about different sort of levels in the central nervous system, from the spinal cord through the brainstem and then ultimately up to the cortex. And again, I'm thinking this cranial nerves and nuclei might offer us an opportunity for some extra work for students who want a little extra credit. When we look at the central pathway, we will look at the cerebellum and the cerebrum. We'll just kind of leave it there for now. Just recognize these are additional structures in the central nervous system. We went through the different construction of the nerve fibers. We talked about axons. We talked about dendritic surfaces. We know there are going to be 12 cranial nerves that you'll ultimately be responsible for. There are also a bunch of spinal nerves. Unlike the cranial nerves, these don't really have names. These are just pretty much numbered. But there are 4 different kinds of nerve tissue bodies or axon projections whose names I want you to remember. So I didn't really say that very well, so let me just tell you what I'm expecting here. There are these 4 different kinds of structures. Some are found in the peripheral nervous system, some are found in the central nervous system. I mentioned earlier the spiral ganglion, didn't I? That's right here. That's this collection of cell bodies, and since it's in the cochlea, it's not part of the central nervous system. This is still out in the peripheral nervous system. The central nervous system doesn't start till you get to the spinal cord, the brainstem, or the cortex. And so a ganglion, many of them together being a ganglia, being not a ganglia, but ganglia, is a collection of cell bodies in the peripheral nervous system. So where here we have this swelling of nerve tissue that contains a collection of cell bodies, that is called a ganglion. If there were a bunch of them, they would be ganglia. Got it? So collection of cell bodies in the peripheral nervous system, ganglion, or ganglia is plural. What if you have that same collection of cell bodies in the central nervous system? Then it's called a nucleus. I'm sorry that it's the same word as the nucleus of a cell, but here nucleus refers to a collection of cell bodies in the central nervous system. You can think of these as being like relay stations or kind of like processing points, processing centers where activity will be summed across different neurons, and the objective being facilitating processing, facilitating sensory and perceptual events in areas higher up in the central nerve system. The thalamus is an example of a nucleus. There are some cochlear nuclei, and again, we're going to go through a lot more examples of these later on. A tract is a collection of axons in the central nervous system. So if we have, again, a trunk of nerve, of axonal material traveling through the central nervous system, think now of your spinal cord, that would be a tract. That would be a collection of axons, again, in the central nervous system. And if you have that same collection of axons in the peripheral nervous system, it's called a nerve. So if we put it all together here, here's the cochlear nerve, here's the acoustic nerve, the vestibulocochlear nerve. Here's that spiral ganglion. Here are the hair cells, of course. So out here it's a nerve. Once it gets in here, once it gets into the central nervous system, it becomes a tract. All right, we have a ganglion out here, a collection of cell bodies out here. Once these get into the central nervous system, now they're called a nucleus. So, ganglion, nucleus, again, collections of cell bodies either in the peripheral nervous system or the central nervous system, and then nerve or tract, depending again on the location. Collection of axonal material out here in the peripheral nervous system, or collection of axonal material here in the central nervous system. By the way, this is a very nice little diagram of the central auditory pathway, and again, in a week or two, we will be looking much more closely at that. Thanks.